Hello learners, welcome to the session. Today's topic is the types of optical microscopy. So this is the topic of the cell biology course in the B.Sc Honors Biochemistry program. And this is the topic is also a course Bioanalytical Techniques, which is the course of the MSc Biochemistry program. So both programs are on offer. So today I will discuss about the what is microscope, what are the techniques of the microscopy. So the topics will be covered in this session are introduction to principal and basic parts of microscope, what are the basic terminology used in the microscopy, and describe the working principle, basic parts and application of the following microscope like bright field and dark field microscope, phase contrast microscope, fluorescence microscope and confocal microscope. And you can also identify staining dyes and fluorescent dyes and their use in the microscopy. So this is the topic of the scientific study, especially to the microscopy. Microscopy is the technique that used for the visualization of the smaller things. So the study of invisible things like cells, viruses, bacteria, eukaryotic cells, plant cell, animal cells is possible with the use of the microscope, because in our world there are many things we cannot see through our eyes. So we need to a instrument like microscope that enable us to see the little things that cannot be seen by human eye. So today in the science, especially in the biochemistry, microbiology, biotechnology, we use the microscope. So microscope is an important tool or techniques used in the labs. So when we are going to see the little bacteria or viruses or the human cells, we need to use the microscope. So in this session I am going to detail of the types of microscope. So there are many types of the microscope used in the biological sciences. So basically here we discuss about the light microscopy. Basically light microscope is used to the light and the optical lenses. So this is the combination of the light and optical lenses to visualize of the biological specimen. Biological specimen can be the source of the biological specimen like the plant cells, animal cells, bacteria and viruses, and other things like the non-living things is also. So this is the relevant topic of the microscopy in the biological sciences. So basic purpose of microscopy is to visualize the invisible things that are too smaller, and to magnify the object, and to increase the resolving power, to improve the clarity and details of image, and to examine the finer detail of the objects. So this is the basic purpose, to use the microscope to visualize of the smaller things. And in our surrounding there are so many bacteria present here, but we cannot see. They can cause the many diseases to the human beings and the animals. So when we want to study of that type of the bacteria, we need a microscope. We cannot see through human eye these smaller things of the microorganism. So in previous session I have discussed about the principle of the microscope, basic parts of the microscope, if you recall the previous session. And here I will give the overview of the parts of the compound microscope. Basically compound microscope consists of the optical lenses. So basically if you can see this figure of the compound microscope here, so it contains the optical lenses, it contains the condenser lens and the eyepiece. The most important part of this compound microscope is the light source, objective lens and the eyepiece. So these are the things that can make the smaller image in the microscope that can be seen by the human eye. So the compound microscope enables us to visualize the smaller things in bigger size. So here you can see the light passes through the condenser lens, and the condenser lens focus the light onto the specimen, where we put the specimen under the microscope, and the objective lens capture the light comes from the specimen. So objective lens is the main lens that magnify of the smaller specimen, and then objective lens release the light onto the eyepiece. So the eyepiece and the objective lens both are produce the magnified image on the eyepiece that can be seen by the human eye. So this is the basic pathway of the light that can produce image under the light microscope. And apart from the optical lens, eyepiece and the condenser lens, there are some adjusting knobs here. You can see coarse focus knob, fine focus knob and stage adjustment knob that can use for the setting of the specimen slide on the stage of the microscope, where we put the microscopic slide in the microscope for observation. So basically the principal microscopy is determined by the resolution and the magnification. So these are two terms basically used in the microscope. Magnification means to view of the smaller things in larger way. So it is the ratio of the image of the large image of the object in relation to the size of the object. So and the resolution it means the clarity of the image. So we can resolve the image under the microscope using the optical lenses. Basically resolution or resolving power to see the closely points as a separate entity. It is the resolution, and the resolving power is the ability of optical lenses or objective lens to see clearly two entities as a separate entities. So it is the resolving power here. So these are two terms, magnification and resolution, are used in the microscopy technique. So now I come to what are the types of light microscope. As the advancement of the microscopy, there are different type of microscope used for different purposes. So here we discuss about the 5 types of microscope, like bright field microscope, dark field microscope, phase contrast microscope, fluorescence microscope and confocal microscopes. So these are 5 types of microscope are widely used in the teaching and research field of the biochemistry, microbiology and biotechnology. So basically the bright field microscope is the microscope which are routinely used in the biology lab. So bright field microscope produce a dark image of the object against a bright background, as its name bright field microscope. So the image is dark appear on the bright background. It means dark image appear on the bright background. So here the bright field microscope utilize the staining specimen because it produces the contrast image under the bright field microscope. So a specimen to be viewed should be stained. When the light passes through the light sources, it focus on the condenser lens, and condenser lens again focus this light, transmitted light onto the specimen, and then specimen interact with the light and it further goes to the objective lens. So objective lens capture the light, reflected light, and then passes to the eyepiece. So bright images appear against dark images appear against the bright images and bright background. So here is image is formed by undifracted light interfering with diffracted light from the specimen, and less transparent part of the sample would therefore appear dark against the bright field background. So bright field microscope is characterized by the dark image against the bright background. So you can see here some of the images of the bright field microscope captured. The bacteria shows the dark image against the bright background, and these are the images of the plant cells which are stem cells. You can see here these are the stem cells. So basically the bright field microscope require the fixation of the sample and then staining dye. So stain is required for biological sample if we want to increase the contrast image. So this is the bright field microscope. This is the light path. This is the light source here, and the condenser lens. Light goes to the condenser lens from the light source, and it interacts with the specimen, and then objective lens, and then goes to the eyepiece. Then eyepiece and objective lens makes the total magnification. If we use the magnification of object lens 40x and eyepiece is generally of 10x, it is the total magnification of the image 100x. So this is the bright field microscope. So bright field microscope is simple to use. It is generally present in the biological lab for the identification of the microscope, to view of the microorganism cells, and to view the morphological study of the cells, and we can use also fixed instant biological samples. So generally in biology, biochemistry labs, these microscopes are used for the visualization of the microorganism of the cells, because some of the cells are stent, some of the not. So some common stents we use for visualization of the samples, like biological components like hematoxylin stent dye used for the nucleus. It produces the purple color. And eosin dye used for staining of the cytoplasm and plasma membrane, and it produces the pink to red color. Methylene blue is one of the dye that used for stain cell nucleus and bacteria. It produces the blue color. So toluene blue is used for the nucleus of plant cell. It produces blue color. Iodine used for xylem and starch content plant cell. It produces the blue and black images. Leishman, Giemsa, right stain used for blood cell analysis. It is good stain, Leishman and Giemsa stain, for identification of different types of the blood cells like RBC, WBC, eosinophil, neutrophils. So it produces the violet color. And safranine used for the plant cell nucleus. So it produces the red color. So these are different kind of the staining are used to increase the contrast image of the biological specimen. And those biological specimens are not stained with the staining dye, then we use the dark field microscopy. So dark field microscopy is another microscopic technique that produces the bright image against the dark background. So this dark field microscopy contains the specific condenser lens equipped with the dark field stop to block the light source before entering into the condenser lens. What we do in this light microscopy, the setup of the light microscope is that when the light comes from the light source, we put the dark stop. Basically we block the some of the light before the condenser lens. In between the light source and the condenser lens, there is a dark field stop. You can see in this image the light is blocked, and after the dark field stop, the light goes to the condenser lens as a hollow cone. So uniform beam of the light fall on the specimen stage and then goes to the objective lens and then eyepiece. What will happen here, that when the light comes from the condenser lens and interact with the specimen stage, light produce the scattered. It means light are produced in a scattered way. It means when the light interact with the specimen scattered the light. So scattered light is captured by the objective lens. It goes to the eyepiece and then it produces the bright image against the dark background. So this is the basic working principle of dark field microscopy, which are used for those objects that are not stained in the bright field microscope. So these bright field microscope and dark field microscope both are applicable in the study of the smaller microorganism of the cells. Here you can see the bacteria, and the most important thing here, in dark field microscopy there is no need of the staining dye, so or the fixation of the cells. So we can view the cells in a life stage, a living stage. So here you can see the bacteria image. In this here you can see the bright image of the bacteria against the dark background here. So you can also some like the plant spore here. You can see here they are showing the bright image. So we can see this kind of the biological specimen under the light field microscopes. So light field microscope is applicable for observing the flagella on the surface of the bacteria and protozoa. It is widely used for examination of the various biological species like algae, plankton, diatoms, insect, fibers, yeast and protozoa, and etc. So this dark field microscope is employed to examine mounted cells and tissue. This is the dark field microscope. And the basic setup of the bright field microscope or the dark field microscope are the same, but differences is that we put the dark field stopper page in the dark field microscope. Now come to the phase contrast microscope. Phase contrast microscopes is produce the contrast image. because it is the advanced form of the bright field or the dark field microscopy. We can say this is the modified form of the microscope, which is produce the contrast image with high resolving power. So here the phase contrast microscope has the specific objective and the condenser lens here with the phase plate. So Fritz-Zernike, a Dutch physicist, developed the phase contrast microscope in 1934. So this microscope is used to improve the contrast images. Basically, contrast images are produced resulting from the differences between the cells and the surrounding medium. So phase contrast microscope used to view colorless and transparent living cells or microorganisms. So the most important things is that we can visualize the biological entities in living state. And the phase contrast microscope is based on the phase changes in the refractive index of light, producing a contrast image of the specimen. What does it mean? That it means that there is phase changes because of the light, because of the refractive index of the light. Here in phase contrast microscope, there is a phase change because of the light interaction with the different medium of the biological specimen. If we talk about the cell structure contain the different kind of the organelles like mitochondria, Golgi body, chromosomes, like chromosome in the nucleus and endoplasmic reticulum. So when the light interact with the different medium of the cellular structure, light velocity is changes. So there is a phase changes here in the phase contrast microscopy. Utilizing the phase changes, a phase shift in the phase contrast microscope produce the contrast image as compared to bright field and dark field microscopy. So what are the main parts of the phase contrast microscope are light source. Generally in phase contrast microscope, light source is halogen lamp or LED light is used, and the two specific types of the condenser lens and objective lens are used. They are equipped with the phase annulus, or we can say annual ring. So the annual ring is just below under the condenser lens. It is a circular disc that contains a ring-shaped groove that allow light to pass through the trinocular head of the phase contrast microscope. Another important parts of this phase contrast microscope is the objective lens with phase plate. So the phase plate क्या करती है? The phase plate distinguish between the light that is not deviated and the light that is diffracted. So here you can see here the working of the phase contrast microscope. The basic principle behind the phase contrast microscope, that detection of the phase changes in the refractive index of light between the cells and the surrounding medium. So here you can see the light sources. The light source produce the light here, and the light goes to the annual ring. Annual ring has a small hole that through the light passes through the annual ring and it goes to the condenser lens, and condenser lens produce further condense the light and focus on the light onto the specimen, biological specimen, which to be observed by the phase contrast microscope. So when light interact with the specimen, here the different kind of the different media within the specimen. Just we take an example of the cell structure. So cell structure, inside the structure there are so many medium here. Cytoplasm is also there, nucleoplasm is also there, the different kind of bacteria, endoplasmic reticulum, Golgi body, mitochondria and lysosomes and the nucleus. So when light undergoes to this type of the structure, light radiation interact and comes after interaction, the light velocity changes. So some of the light is slow, and when it light slow, and the light basically bends, the phase shift. So in this in that phase contrast microscope, the phase contrast microscope produce two types of light. one light is diffracted from the biological specimen, which produce the changes in the refractive index, and one light are not deviated. Just a non-deviated light just passes through the surrounding of the biological specimen. So this type of the light is captured by the objective lens, and objective lens release this the light which are deviated from the specimen and goes to the phase plate. So here the phase plate differentiate between the phase shift of the refractiveness of light, and it further goes to the primary image on the eyepiece. So when the diffractive light and the non-diffractive light recombine again, it produces the contrast image under the phase contrast microscope. Here you can see the illuminated light, diffracted light, undifracted light, and the combined diffracted and the undifracted light. So basically the phase contrast microscope is the microscope that detects the changes in the refractive index between the differences between the surrounding medium and the cells, and this is the result of the producing of the contrast microscope. Here you can see the path of light. Here light source goes to the condenser with annulus, and then specimen, and then objective, and then phase plate with annulus and the image plan. So here you can see the image of the phase contrast microscope. Here the gray image is produced as a contrast image under the phase contrast microscope. So gray background with the light and dark which are found across the sample. So different kind of the density of the specimen interact with the light, and light intensity, the velocity changes. So there is phase shift here that change and the captured by the phase contrast microscope, because as its name phase contrast microscope, the phase changes and it produces the contrast image under the microscope. So the important things is that phase contrast microscope produce the contrast image. It does not need to staining of the sample. We can see the biological sample, those are not stained, and we can see the biological object that are colorless and unstained. So this is the basic helpful phase contrast microscope features. The application of phase contrast microscope is we can view unstained specimen or cells, examination of growth, behavior, morphology of living cells in vitro cell culture. The phase contrast microscope is widely used in animal tissue culture, and we can how cell grow in the cell culture medium. We can observe every day to see the morphological features of the cells, and visualization of intracellular structure in blood cells. We can see the mitochondria, endoplasmic reticulum, what are the internal structure of the cytoplasm, and the other eukaryotic cell structure, the cell membrane. We can visualize by the phase contrast microscope, and we can detect abnormal growth of cell in disease diagnosis. If the cell's growth is abnormal, so it indicates some internal changes because of the uncontrolled growth. So we can detect the cancer disease also if we culture the cancer cell in the animal tissue culture lab. And highly transparent organism can be seen and determine the motility of microorganism. We can see the flagella movement on the bacterial organism. So this is the most important phase contrast microscope. Basically, this is the microscope is used in the research lab. And again, another microscope is the fluorescence microscope. As its name, fluorescence microscope, it uses the fluorescent dye to view of the biological specimen. So this fluorescence microscope is an important scientific tool in biochemistry and biotechnology and microbiology. It is based on the imaging of the fluorescent labeled specimen. It means a specimen is fixed with the fluorescent dye first. Before observing of the specimen under the fluorescence microscope, we generally fix the cells or stain the cells with the fluorescent dye. So there are different kind of the fluorescent dye used in the fluorescence microscopy. So it uses the fluorescent dye to study scientific molecular structure and cellular components. So we can identify the specific kind of the molecules like protein or the DNA using the fluorescent dye, and under the fluorescence microscopy. And in this fluorescence microscopy, we expose a specimen to ultraviolet light or the blue light, because the UV light or violet light have the shorter wavelength to excite of the molecules, and fluorescent dye is used to stain the specimen, and it is called the fluorescence microscope. So you can see the setup of the fluorescence microscope here. So what are the basic part of the fluorescence microscope is the mercury lamp, which is the light source here. It contains 3 types of the filters that can detect the excitation of light. Like the excitation filter is used to select only desired wavelength of light, which is the excited, comes from the mercury lamp. And another mirror is the dichroic mirror. So it is an optical device that reflects the excitation light from the objective to the sample. Another filter is the emission filter. Emission filter eliminates any excitation light that reduce the contrast of the image. So the whole setup of the basic component of the fluorescence microscope inbuilt in the instrument of the fluorescence microscope. How fluorescence microscope is work is that when light produced by the light source like mercury lamp, only the excitation filter detect the light that are excited. When these excited light fall on the biological sample, which are already stained, fluorescently labeled stain, that the fluorescent labeled stain specimen absorb the specific wavelength of light and produce the higher wavelength of light. And this higher wavelength of light captured by the dichroic mirror, and the emission filter capture the emitted light and goes to the human eye. Or there is a detector which attached to the CCTV camera, and this image can be seen on the computer screen. So here the basic fluorescence microscopy principle is this depend on the Stochastic. It is the capturing of the higher wavelength of light. What will happen during the fluorescence phenomena? Fluorescence is a phenomena. So fluorescence occurs when fluorochromes absorb certain wavelength of light, and the difference between absorption and emission spectra is called the Stochastic. So there is a differences between absorption spectra and the emission spectra. So when the fluorescent dye absorbs the specific wavelength of light, it goes to the electrons of the molecules of the fluorescent dye, move to the excited state, and at this excited state comes back in the ground state. So in between there is a photons emitted from the moving of the electron from excited state to ground state. So this difference between the absorption spectrum and the emission spectrum, so this difference is called the Stochastic. So the emission of the higher wavelength of light is captured by the fluorescent microscope that visualize the fluorescent labeled specimen. Basically it is used for detecting the specific molecules, live cell imaging, and study of antigen antibody interactions. So here you can see the figure is the Stochastic. This is the blue line is showing the absorption light, and the green line is showing the emission light. So there is a difference between the wavelength of the two absorption light and the emission light. It means the two different kind of wavelength here. So the fluorescence microscopy is captured by the capture of the higher wavelength of light comes from the fluorescent labeled specimen. So what are the fluorochromes? Fluorochromes are the specific dye or compound which are fluorescence characteristic features. So here are some of the dye. You can see fluorescent dye, host 3342, DAPI, ethidium bromide, echidna orange, stocks, green, propium iodide, fluorescent, EGFP, M cherry. So these kind of the fluorescent dye have the excitation wavelength and the emission wavelength here. So I hope you have understood this type of the microscope, basic parts and basic principle in this session. Thank you.
Do you wish to be part of a television studio like this or any other media system? Or already part of any media system, want to enhance a new set of skills? Welcome to School of Journalism and New Media Studies. Here in the School of Journalism and New Media Studies, we are offering program for all set of people to learn and enhance the media skills. The vision of School of Journalism and New Media Studies is to generate a new set of professionals in the area of media and communication studies with understanding about role of media in the national development and global understanding. We offer innovative programs in the field of journalism and mass communication that sets the benchmark of high quality learning and teaching process at the national level. We offer these programs across the country with the help of our regional network as well as study center. Same manner, many of our programs